Hi guys, Anna is here, and in this A-Level Biology Masterclass, I cover all of the topics in 5 minutes. So if you want to learn biology syllabus in a simple and efficient manner, then please subscribe to my channel. In this video, we're going to discuss the structure of phagocytes and the process of phagocytosis. So if you didn't know the difference between neutrophils and macrophages and the role they play in the primary immunity, then this video is for you. So I already started writing out here, and you can see the two major phagocyte types that you guys need to know are the neutrophils and the macrophages. In exams, you can be actually asked to draw out the neutrophils or the macrophages in a 2 or 3 marker question, so it's important to be able to differentiate them. So the neutrophils are specific in the sense that they have this multi-lobed nucleus, and in their cytoplasm there will be loads of granules, which will contain different molecules and digestive enzymes. So this is the way to draw a neutrophil. A macrophage is very similar, however, its nucleus is in the shape of a kidney, so we're going to call it kidney-shaped nucleus, and you're supposed to draw it like that, and there will be no granules present in the cytoplasm. So as long as you guys know how to draw this and differentiate them, then you're absolutely fine. Neutrophils and macrophages also have different features. So for example, macrophages are long-lived cells, however, neutrophils are short-lived cells. And for example, neutrophils have this ability to squeeze between the capillary walls so they can circulate and reach the organs. And they do that by the process of chemotaxis. So chemotaxis is when the neutrophils get attracted to a site where the pathogens are by the chemicals released by pathogens. And then also neutrophils constitute of about 50 to 70% of circulating blood cells compared to the macrophages. The neutrophils also mature in the bone marrow versus macrophages maturing in the tissues. And they only make up 2 to 8% of circulating white blood cells. So I've written all the points here, guys. If you copy those down, and just that helps you to differentiate between the two. Now let's move to the process of phagocytosis and see what the neutrophils and macrophages actually do to conduct this process. So I'm going to draw an overall process, and then we're going to subdivide it into neutrophils and macrophages functions. So first of all, a phagocyte, either neutrophil or macrophage, doesn't matter at this stage, will approach a pathogen, which we'll mark here at the star and label it so. Okay. So the key word you need to use in your exam is that the phagocyte engulfs a pathogen. And the key thing is here, guys, when it does engulf a pathogen, the pathogen will be engulfed in a structure called a phagosome, which otherwise can be called phagocytic vacuole, so which I'm going to label here in a moment. The whole point of having a phagosome and containing a pathogen inside of the phagosome is because in the next stage now we're going to fuse phagosome with a lysosome. And remember, guys, lysosome is a type of an organelle that contains digestive enzymes. So for a phagocyte to be able to digest a pathogen, what is required is a separate environment of where the digestive enzymes can act. And this is what is provided by the phagosome. And when the phagosome fuses together with the lysosome, they make a new structure, which is called phagolysosome. So basically, digestion of the pathogen can happen in a separate environment, and the digestive enzymes are contained within the phagolysosome. Otherwise, if they would be around the cytoplasm, they would be digesting separate parts of the cell. Right, so up to now, the pathway of phagocytosis is very similar for neutrophil and macrophage for the purposes of A-level biology. But the final stages of phagocytosis are going to vary between the neutrophils and macrophage, and this is what's going to happen now. Okay, so let's separate them into two, and we're going to have neutrophils here at the above. When the neutrophil digests the pathogen, it completely digests it, and this results in the death of neutrophils. So neutrophils die after this digestion, and eventually the neutrophils will form a pus. This is PUS for the spelling. And pus in molecular biology and immunology is a sign of dead neutrophils. Right, let's move to the macrophages now. So the digestion which is carried out by macrophages is very similar. However, the difference is that the macrophages, they do not digest the pathogens fully, and instead they will be saving of some antigens and they will be presenting them on the major histocompatibility complex, which is abbreviated as MHCs. And those are the receptors that are presented on the macrophages, on their cell surface membrane, and then the antigens are going to be presented on the MHCs. And this binding of the antigen to MHC makes macrophages as antigen-presenting cells, which is abbreviated to APCs. And as a result, when the antigen is presented to the B cells and T lymphocytes, it will result in initiation of primary immunity. And this is, guys, everything for my phagocytosis video. Hopefully you now know the difference between the neutrophils and the macrophages. Please press the thumbs up for my video, and I will see you in my next video. Goodbye.